Welcome back to another episode of the DNA of Things, where we are building a community of conscious creators. I'm so grateful to have you here today, where this time we are joined with Dr. Carlos Bustamante, a renowned expert in population genetics, whose work spans continents and cultures. Now, from his early days growing up in Venezuela to the groundbreaking research at prestigious institutions like Cornell, Stanford, Dr. Bustamante has been on the forefront of DNA sequencing and genomics. In this episode, we'll explore his journey and his role in advancing the field of personalized medicine through his initiative, Galatea Bio. Now, this venture is not just about genetic testing, it's revolutionizing our approach to incorporating global genetic diversity into precision medicine and developing therapies that cater to unique genetic profiles. Dr. Bustamante will share insights into the creation of this Galatea Bio Passport, a tool designed to personalize healthcare by providing detailed genetic profiles, including ancestry and potential health risks. Now, join us as we unpack the complexities of genomic medicine and the importance of diversity in genetic research, aiming to make cutting-edge genomic insights accessible to all. This is the future, and we are creating it. Let's dive in. Dr. Carlos Bustamante, thank you so much for joining us today.
It's my pleasure to be here, Jeremy. It's super fun to hear about the DNA of everything. Is that right? Or the DNA of things, or
DNA of
the
things,
DNA of whatever. The DNA of things, like the Internet of Things, but the DNA of things. I love it.
Yeah. Well, I mean, in a monumental day, we were just talking about the solar eclipse that we just kind of passed through, and we won't pass through again until 2045. So imagine where we'll be at that point in time. So, hey, Carlos, there's like a ton of stuff that I know we're going to dive into today in the realm of, Galatea, the future of, let's say, the DNA of things, like the DNA, biobanks and precision medicine. I think a lot of the work that you're doing has parallels with some of the insights we're starting to see from the All of Us projects and many of these other national data banks of DNA that are starting to build up. And so I suspect you have a ton of insight to speak to. But before we dive in,
at least some strongly held opinions. So
right, well, that's what. Well, perfect. You've come to the right place. Before we dive in, I'd love to hear about the man behind the mission. So tell me about, you hail from Venezuela. Tell me about
I'm originally from Venezuela.
Yeah.
So I was born at a time when Venezuela was probably the second or third richest country in the Americas, right? Certainly after the US and Canada, it was a close toss-up. And I just had a really unbelievably eclectic childhood. So my dad was is a physician, and when he graduated from medical school, they sent us to a small island off the coast of Venezuela called Margarita Island, and he was like the island doc, and he got paid in chickens and whiskey.
Wow.
And I had an unbelievably cool childhood living, in nature and on the beach and growing up in a really idyllic setting. And both of my grandparents were very influential, and from an early age, I was always reading. And one of the things I remember reading, probably I was 4 or 5 years old, were these comic books, but they were about history and evolution. And so I was interested in evolution and human history probably since I have early memories, right? So it was a really interesting, way to kind of frame what ended up being my life's passion. But I have a collection of books from when I was, 5, 6 years old that are all around each of these. And it was a series, it was like a serialized cartoon. And so like every week was one, like Cro-Magnon was one week, Neanderthals were another week, the Egyptians were one week, the Romans, the Greeks were a different week. So like I kind of had a classic education in that realm, and little would I have suspected that it's been the better part of 25 years sequencing populations to understand the historical record tied into the DNA of people and how we might be able to understand the great human diasporas from DNA sequencing. So always very interested in science, always very interested in history, very interested in anthropology, and ended up running a lab at Stanford that we used to call the Gonzo Genomics Lab, and it was a little bit like Andy Warhol's studio. We just had all these cool mathematicians and anthropologists and physicians and people coming through working on cool stuff. So people would ask me, what does the Bustamante lab work on? And I was like, oh, it's whatever Bustamante is interested in. And so people would just pitch me cool projects, and we had enough kind of slush money around to take on cool projects. And, so we got to sequence, for example, the genome of Otzi the Iceman at one point, and analyze Otzi's genome and compare him to different populations that we had studied before. We got to work on the genetics of blonde hair in the Solomon Islands. We sequenced
that was you
cool
guys.
genomes.
I didn't know that was you guys.
Yeah.
Oh, wow.
Yeah.
Wow.
Yeah.
Very cool.
So we did that.
But before Stanford, you were at Cornell, weren't you?
I was. Yeah. So I was, again, sort of in the agricultural genomics world and different genomes. So
what years were you there?
I was at Cornell from 2002 through 2010.
Wow. So I just missed you. I came there in 2010. Yeah.
Yeah. We were like two ships passing in the night.
That's right. So you must have done some work with Sean Niles, the Apple guy, Apple Genetics.
Oh, yeah.
Yeah.
Sean Niles was my postdoc. So Sean, as I tell you the story, Sean came to me and he had done his PhD in the Solomon Islands. And he said, I want to go back and do a postdoc in the Solomons and study blonde hair. I'm like, yeah, bro, I'd like to go do a postdoc in the Solomons too, man. That must be cool. And he said, I think they got blonde hair for a different reason than you think. And I'm like, nah, that's a Captain Cook allele. I can tell you why they got blonde hair. Luckily, Sean was very persistent in that, Canadian way, very polite, but very persistent,
Right.
And so he got funding and went out to the Solomons and worked out all the details of how we would consent people and do the study. And he went over all the islands, and he came back with 1,000 samples. And it was an unbelievable, just knocked it out of the park kind of project. He teamed up with Emer Kenny, who was a postdoc who had just landed in the lab. And I like to say that Emer had the best week anyone had in the lab, because we'd just gotten the data. And within 3-4 days, we had this whopping signal over the gene TERP1, which turned out to be the gene for blonde hair in the Solomons, but not the gene for blonde hair in Europe, right? They have, blonde hair because of OCA2 or KIT ligand, not because of TERP1. So it really got me thinking, like, if that's blonde hair, which you don't need to be a Stanford trained dermatologist to phenotype. You got to be about 6 years old and like, oh, I wonder why some people are blonde, why they're redheads, why, what's up with that, And there's two populations that have blonde hair, and they have different genetic basis for blonde hair. Why would you think diabetes has the same genetic basis all over the planet when we know it presents differently, right?
Right. Well, yeah, we know that about
really got us thinking about this.
Yeah. It's like the lactase persistence genes, right? There's multiple polymorphisms there. Yeah.
There you go. And same gene, different mutations, right? So the Maasai have a totally different homegrown mutation than Northern Europeans. So my guess is it's the rule rather than the exception, right? Evolution finds a way.
Well, that's interesting. So like you're here on like the tip of the spear in terms of population genetics. And I want to, spend a good amount of time talking about Galatea, because that's some pretty exciting stuff that you got going on. Like you just talked about Sean Miles, and yeah,
MYLES. Yeah.
that's right. Yeah. Sean, and his, his feed of the day, 1,000 samples, like that was considered a lot, I think, at
That
the
was
time. What?
huge. That was
Yeah. 15 years
that
ago.
was a big deal. That was a
Yeah.
big deal.
Yeah. And so just with that small amount of sampling already, there's like a paradigm shift in like, oh, maybe there are multiple origins of things in our, human library of phenotypes, like for listeners who are not evolutionary biologists, and I'm going to just, put a whole disclaimer up front. I'm a huge Stephen Jay Gould fan. And so when I hear you start to talk about,
so I taught evolution for Gould when I was a PhD student.
there you go. Right. And what a great fit that must've been, because he is so dialed into the great books, the great class. I actually learned, J. Adler Mortimer's suite of the Great Conversation. So this is like the great books from Homer's Iliad up to Einstein's theory of relativity and beyond. Works really kind of fizzled out past works in the 1950s, because this was only written in the 70s. But how do you know something is great, if it hasn't survived the test of time? So there's definitely more books that need to be added. But I was delighted to see,
I would put ontogeny and phylogeny. You could add to that list.
Yeah, it's actually propping up my computer right now, believe it or not.
No, it was crazy because I was a student. So when I landed as an undergrad, I knew I wanted to do population genetics. So I landed to the Science Center at Harvard, and I said, I'm looking for a guy named Richard Lewington.
Oh, come on.
This guy said to me, oh, I'm his graduate student. And I'm like, the alien, take me to your leader. So I went, and that day introduced myself to Lewington and said, hey, look, I'm interested in population genetics. And Dick had this ability to take everybody super seriously, especially if you were not like a professor, right? If you're, it would be his perfect thing to take some random undergrad who was interested in evolution and give him 2 hours of his time. And that's what he did. And so I started working in the Lewington lab, and it was an unbelievable time because this was before genomics. This was, Lewington was criticizing the Human Genome Project, writing books like Not in Our Genes. And here we were trying to get at, models of evolution and how you would test them using statistics. I mean, that was kind of the theme, and so I took his course on evolution, and Gould had been on sabbatical that year. And then the following year, so Lewington was driving down, his lab. He had one grad student left or two grad students left. And he said to me, well, look, I need a TA. You've taken the class. Do you want a TA for me and Gould? And I'm like, yes, like, absolutely. So I TA'd them for like 6-7 years until they wound down the class. And it was unbelievable because, it was a master class on the contrarian views of evolution. Like everybody had, you had to go into the class already having a working notion of Darwinism because they would tear it down. That was their whole goal, right? To resolve it nearer to their own view. But, population genetics back then was theory rich, data poor. We thought we understood everything. To your point about Einstein, it was a lot like 19th century physics. Like all we got to do is solve this black body radiation problem, and then everything gets worked out. And then it all exploded. I mean, Lewington said to me when I went to go start my PhD, and I said, oh, I want to do theoretical population genetics and I want to study the statistics of DNA. He said, Carlos, come on. No place is ever going to want two professors that work on the statistics of DNA. It's just too niche a field. So even Lewington, like, it was unforeseen, right? Then Greg Venter raced the Genome Project, and Francis, and wouldn't be beat. And so the two of them raced to the end and accelerated what was going to take 10 years. It got done in 3-4 years. I mean, it was an unbelievable time. It was an unbelievable time.
That was an interesting co-opetition time. What would you say? Where I'm not sure that Venter could have got his project done, leveraging the shotgun sequencing approach, if he had not had the backbone, like kind of contiguous array that was put together
No,
on the nature
I know
side
it
of
was
things.
interesting.
Yeah. Mm-hmm.
It was what was also very cool for me was so Solera, right? Here's this private company started by a guy that used to be at NIH because Venture was at NIH. That's where he had
That's
started.
right.
He used to be at Genome Research. And
Right.
the Aplera was the parent company to Solera and ABI. And ABI had like the best business model in the world, right? It's like, I'm going to sell machines to our own subsidiary. And then the federal government came in and said, you can't do that alone. For every machine you sell your subsidiary, you got to sell the government one.
Wow.
Okay.
Tough job.
I'll take that deal.
Wow.
I'll take that deal all day long.
Wow.
And so,
yeah. Go ahead. Sorry.
But then Solera ended up in a bubble, the biotech bubble, which followed the internet bubble, and then went belly up, right? They went bankrupt. And so not a lot of people know this. We ended up with all the data at Cornell. So that the first papers that made my career were papers where we had the Aplera data. We had exome data in 2002, 2003, the first 40 exomes that ever got sequenced. We had it at Cornell. It was Andy Clark and Rasmus Nielsen and I, and we had an unbelievable time. Like we were building new models, we had access to cool data. And that was the first time I thought, industry can do things at scale. That's hard, almost impossible for academics to do on their own, even for government, because it's got to move with everybody, right? Like when you do these big government projects, like you can't just run ahead. You've got to bring everybody along, right? It's the right thing to do. Yeah. So it was a really interesting learning because we became known as the lab to send distressed data assets to, either on the government side or on the industry side. And that's what we did for a long time, like just bioinformatics of big data that required some data cleaning. And then we just, brought cool models to bear on the data.
Interesting. So Solera, Craig Venter's startup, that was focused on human whole genome sequencing, like providing that. Is that correct?
Well, they did Craig's genome, and then they did 39 other genomes, but they didn't do them as genomes. They did them as exomes because it was still like unbelievably expensive.
Right.
So they spent a couple $1 million per exome.
Right. I think was Steve Jobs in and amongst those? I remember hearing rumor that he was, or you can't say.
I'm not gonna violate Steve Jobs' HIPAA, man.
Right. That's right. Yeah. I redact the question. So Solera was obviously a private entity. When he did the Sargasso Sea, was that a private initiative also, or was that still government?
I think by that time he left Solera. He'd cashed out. He was on his sailboat. Was on to greener pasture, bluer oceans. You choose your Sargasso Sea is a mixture of green pastures and blue oceans.
Right. Yeah. Interesting. Okay. So what a great springboard into Galatea, right? Like, cause we've just kind of, for, let's just say the non evolutionary biologist / big data genomicists, we just kind of talk through the last quarter century of history where, if you think about, pre-2000, we had the odd gene sequenced, as measured or as quantified by PCR, which, by the way, probably everybody knows PCR now because of post-COVID. And so that was a mechanism of sequencing gene fragments. We had microarray data that would tell you which variant of a gene that you have. But now, because of all of this, work that you've pioneered, frankly, and in the confluence of industry, we can now do something really like unfathomable before, which, is why we can kind of let your predecessors off the hook insofar as not being able to anticipate.
No, I mean,
right.
Like the old saying about standing on the shoulders of giants, right? So our generation of population geneticists had the best of both worlds, right? Well, it's Charles Dickens. It was the best at times. It was the worst at times. We had wonderful theory and lots of hypotheses, and we had no data, right? And now we're living in a world of unbounded data and little theory to guide us, right? Because there are all kinds of funny biases in the data, right? So I spent many years working on evolutionary models that you could only fit if you sequenced, right? And then SNP chip data came out, and it was like, okay, well, let's throw all that out, right? You can't use that in theory because of ascertainment bias. So, like, you got to bring in other stuff like principal component analysis and robust methods that summarize the data without fitting models. But I used to say there are two kinds of people in the world. There are people who like models, and there are people who like summary statistics, and never the twain shall meet, right? And so, today you're living in a world where you're pumping out an unbelievable amount of data,
and really cheap, right? Like, so we're talking like in early 2000s, billions of dollars.
It was $1 million. To get the first. It cost $2 to $3 billion to get to the first genome, and then it cost $100 million to do the second genome, right? So you had a 10x reduction, and now it's human capital and human technician time is probably the biggest contributor to cost. It's no longer even the reagents on the machine. Like, yes, they limit you and the service contract on the machine, but really it's like the sampling and getting the machine in. Like, if you gave me the sequencing for free, it would still be a couple $100 to get the sample in the door. And that's the order of the cost of the reagents to do the sequencing, right? So you're really bandwidth limited on getting samples that are properly consented and have interesting data tied to them, right? Interesting phenotypic data. So the clinical data that travels with the sample, in some sense, is more valuable than the genetic data, because the genetic data alone, doesn't tell you all the story, right? So you need the genetic data plus the phenotypic data in an environment, in a data lake that you can mine and talk between what we would call the geno layer and the pheno layer. Not very Lewintonian too, right? So Lewinton, you remember, you have the genotype space and the phenotype space and the laws of transformation between them. Right. And so we're very interested in that map, right? How do we map? It's very classic genetics. How do you go from genotype to phenotype? How do you predict what something will look like based on its DNA? And then the inverse problem of, based on how something looks, what can you say about the genes underlying those traits, right? So we're very interested in the clinical side of things, medical genetics, and doing sequencing for patients whose clinical record, whose clinical manifestation, whose clinical journey indicates they may have a genetic condition, right? So if you're a woman and you have multiple first degree relatives who've had early breast cancer, and you have early breast cancer, then by God, please get sequenced, because it's important, right? Like that, everything clinically indicates you may, be a carrier of a penetrant BRCA1 and 2 mutation, the breast cancer gene, or more likely some other mutation in some of the other cancer genes, or perhaps even a genetic background, a so-called polygenic risk score that is predisposing you to be presenting in this way. And, breast cancer led the way, then prostate cancer, then Alzheimer's disease, and on and on and on. But, we've now got catalogs that include thousands of traits. I'm not talking 1 or 2 or 5 or 10 or like thousands of traits that we know have some modicum of a genetic basis. And we're just beginning to really wrap our head around those laws of transformation that map us from that DNA level to the clinical level and back, right? And so that whole conversation is mediated by the clinical enterprise, right? What people go to the doctors, how they're seen, how that data is stored, and in digital format today, right? But most of us is an electronic health record. And then the thing that really keeps me up at night is that we pretty much know only about populations from Europe and some from North America, right? That's where this enterprise has been ongoing for the better part of 20 years, right? So
So tell me then, how does Galatea fit into this?
Great question.
Yeah, and I think we've kind of been talking about this on the outset, where we talked about, well, it seems independent origins of various phenotypes from, again,
Yeah.
differing genetic mutations. So, and then we are on this notion of, well, we certainly have a very narrow view of the true diversity that's out there. Yet we're
Right.
sitting on
Right.
this reduction in cost. So enter Galatea.
So in 2011, we wrote a paper in Nature called Genomics for the World. And we sort of threw down this gauntlet and we said, 96% of the participants in these large scale studies are of European descent. We got to do better. And I'd like to tell you, everybody took us seriously and we changed the face of science and it all, worked. And now you have good representation of everybody in the world in large scale genetic databases.
Mm-hmm.
That's not what happened.
Mm-hmm. Mm-hmm.
Business as usual is what happened. And so, and look, let me put something on the record. I love the UK Biobank. I think the UK Biobank is one of the great human resources on the planet and data resources and scientific resources for good, right? We need more databases like the UK Biobank. It's 500,000 people, mostly collected from relatively healthy volunteers, although it was an all-comers cohort, where you followed them longitudinally and seen what happens to them, gotten biological samples, and been able to study the genetics of these traits. So
and I think
it's sort of
there are some twin sets in there also, weren't there?
There's the UK twin study. Then there was the 100,000 K project, which became Genomics England, which was sequencing in the context of clinical care. There's Decode, which again, I love Decode. I think every country should have a Decode. Decode's a country of 300,000 people.
Is that Iceland?
This is Iceland. Decode is built in Iceland. 300,000 people whose DNA has either been sequenced or imputed from those that have been sequenced, because it's one big pedigree. And we have a very detailed understanding of the genetics of rare and not so rare diseases and how genetics contribute to them. And in general, Europe organized itself so that every country, the Finns have a project, the Dutch have a project, and then in the US we have the All of Us cohort. It's our national cohort, and I think we should be proud of it and do what we can to support it. So far, they got a couple 100,000 people in there, more diverse than the other projects. But in a country of 300 million people, 300 million plus, it's a small sampling, first and foremost. Secondly, again, for better or worse, it's a government database. There are lots of people who don't feel comfortable putting their DNA in a government database, and industry doesn't have access to it right now. Whereas the UK Biobank was financed by industry, right? So who's actually going to make new drugs? It's not going to be government. It's going to be industry. It's going to be biotech companies. It's going to be, and that's what you need. It is the prophylactic deployment of medication that can bend the cost curve on the accumulation of morbidity, right? And as the population ages, thankfully, right, we're better at keeping people alive. That's a good thing. Then necessarily you have sicker people, and we need medications to improve their health and ideally deploy the medication so that you can prevent the disease in the first place. And one good example is cholesterol, right? So statins are life-saving, right? You take statins, they lower your bad cholesterol, and the people who take statins reduce, for the whole population, the number of heart attacks and strokes that would otherwise happen, benefiting most of the people that take statins, but they don't have the heart attack or stroke, but then benefiting everybody, right? We don't want people to have heart attacks and strokes. You're losing people at prime prime and into their golden years, right? So we want to make those a thing of the past, and you can you can really trace improvements in cardiovascular disease to reduction in smoking and deployment of statins and other medications. So if it's a big if, but if we want everybody to benefit from these advances in precision medicine, if we want precision health at scale, then we absolutely need to diversify the databases that we use for drawing these conclusions.
Right.
I
And
mean,
so
to me, it's obvious.
just to pull that out is like those statins are not universally, the efficacy is not universal, right? And I think this is coming back to what you were saying at the beginning is, well, there are many potentially many different origins for something like, diabetes. And so when you start to diversify the sample set in addition to all these European data sets, right? Okay, so we're starting to see it in the All of Us project. And then you've got like the South Americas. So tell me about that. How is that going to lead us to this better future? Mm-hmm.
We're trying to build a cohort, which we're calling the Biobank of the Americas. It's really global, right? But we're starting in the Americas, and the goal is any group that contributed to ancestry in the Americas, which is pretty much every group, right? Because they're descendants in the Americas from pretty much everywhere in the world, right? You could almost call it the New York City project,
Right.
or the Toronto project, right?
Mm-hmm.
And sample people from all over the world, the Vancouver project, if you will. And our goal is really to embrace the genetic and phenotypic diversity of our species writ large, right? It is that kind of population genetics dream of getting at every nook and cranny of the gene pool and how it contributes to the beautiful manifestation of what we are like as a species,
Mm-hmm.
So
Mm-hmm.
and we're starting with populations in the Americas for a couple of reasons. Like if you like Iceland, I got 1,000 decodes in the Americas. We got lots of islands. Those islands were formed in the last couple hundred years. And certainly the gene pool has turned over in the last couple hundred years. And so you have the same pockets of founder population, like Newfoundland. Newfoundland's a great example. Founder population. You can study traits, you can make pedigrees. And, the Canadians have known this for a long time. The Montreal work on.
There's a Neiman Pick.
Pick, I think
it is. Yeah.
And the Quebecois and so on. So, we're not doing anything that is radical in terms of the science. If anything, it's radical in terms of its representation. And it's a little bit like if you're thinking about telephony, right? And you could go invest in wiring the places in the world that don't have phones, or you can build cell towers. Let's go build cell towers, right? Or better yet, like let's launch the equivalent of the SpaceX satellite network that gives coverage for internet all over the world, right? Like you can now think about it in that regard, right? So how do we get to a near universal coverage of humanity in terms of their representation in these large-scale databases? And that's our goal. Become, the conduit that creates the world's largest, most diverse database for genopheno studies of both susceptibility to and protection from disease, as well as the use of that data for development of new therapeutic interventions.
And so how long have you been at this now?
We launched the company about 3 years ago. It was spun out of my lab at Stanford. We started with, at the time, an ample set of DNA material, which were used COVID discards. So it turns out you can sequence human DNA from COVID discards. And we got permission from the powers that be to build a study to do this, right? So consenting people so that when they give a COVID test, it said there that we could reuse the material and recontact them in the event they want to be part of other studies. We now added prospective sampling and are now looking for ways to bring more sample types in. So we're basically giving away free DNA kits, finding partners that want to have that material sequenced, returning results to people, a very basic ancestry test, and then if you want a more detailed ancestry test, then we can partner with groups like sequencing.com and others to reanalyze that data and bring more feature content, right? So if you think about media, right, you have on air, over the air network TV, you have cable, you have streaming services, and then you can either choose to have the commercials or pay for premium service where you don't have the commercials, right? So we want to really think of it in terms of media, because it is media in many ways. It's interpretation on top of existing sequencing, and then improving access and literacy, because even doctors don't know what to do with a lot of genetic information. They're busy. They didn't take a lot necessarily genetics in medical school, and certainly the field has changed quickly. So we want to educate physicians. We want to educate the general public. We want to work with people who are hobbyists, right? Some hobbyists know more about this stuff than some experts. Get cool content in their hands so that they can help us vet it, and then work with pharma to put structured projects in place to really lower the cost of collecting genetic data. Because as I said, it's no longer really about the sequencing reagents. It's about getting the samples and getting people consented and getting the word out there,
Well, yeah. And it's amazing just hearing you speak now. And, I love that. I love that frame of the media, because the medium is the message, if you will. And I think about my own personal journey of, genomics, and I'm obviously at a huge advantage because I've been studying it for 25 years. And so when it comes, to the consumer genomics space, like, obviously it's a mixed bag, but you mentioned sequencing.com. And so I have my whole genome there, my wife's and my daughter's. And it's really amazing that, first of all, it's whole genome now, right? It's not just the arrays or a few genes, but then it's also the connectivity to the most recent research. And so, you're basically pinging the research database with all the most recent updates. And I mean, I'll share with you just one that came up in March was highly relevant because, my brother went in, to get some tests, and, there was concern about, different blood cancer metrics, I think is MGUS, and then also the iron tie. And then I get this like update from HealthScan on sequencing saying, oh, I have the variant that's associated with elevated hemochromatosis, for example. And so it's like, oh, well,
yeah, factor 5 Leiden or something.
I'm not sure. But the point was, I could pass that information to my brother and say, hey, okay, well, there's this genetic reason why your iron might also be high. Talk to your doctor. And then it's like, okay, yeah, maybe what we'll do is we'll get you donating blood a couple times a year. Right. So really interesting application, on the precision medicine side, that, also to my daughter as well. Like, I know that I'm a carrier of like a rare genetic disease as it relates to hearing, congenital hearing malformation. And so that was one of the first things I ordered was like the hearing test, because if that's something that is going to be, it's not developed, it's going to delay her, socialization or development of her socialization. So just being able to have that information at different stages in your life phase in the context of, well, this cosmopolitan genome also, right? So you're bringing together all this diversity. Like we don't, I was actually also thinking about this also is I don't really know my health history as it relates to what is heritable, right? There's things that are genetic, there's things that are environmental, and then there are things that are behavioral. I think what you're doing with the DNA is like, well, that's one of the things that from the outset is, oh, well, now I get a true picture of, my family inheritance, let's say, genetically. So okay. So Galatea has been around for 3 years. What's the database look like right now? 3 years.
So we're somewhere between 500,000 to 1 million samples in. We want to
Holy
get
smokes.
to about
Wow.
10. We'd like to get to 10 million.
Yeah.
We are actively recruiting and partnering with academic and health systems in the U.S. and ex-U.S. And we're also doing clinical genetic testing ourselves. So we've got a clinical lab. We do genetic testing, and we're really looking to understand where our thesis holds up and where it doesn't. I mean, there are things that we know are pretty well understood. Pharmacogenetics is a good example, right? We're learning a lot about what genes contribute to adverse drug reaction, and a lot of those alleles can be classified into different metabolic tranches, right? So either individuals who can't metabolize a prodrug because they don't make the enzyme, the protein that transforms it from the prodrug to a drug. Codeine's a good example of that, right? Codeine's a prodrug. If you don't have the right gene to metabolize codeine, you're not going to get analgesic response. Likewise, there's some people that are so-called rapid metabolizers. So if you give them the same dose of codeine that you give, everybody else, then they could actually get an overdose because their body breaks it down very quickly. And then on the other end are people who metabolize it too slowly. So they need, more than you would predict. And so, there it becomes this question of making a map between the genes that are involved in the absorption and metabolism of different medications and what alleles are found in what populations. And we know, for example, there's an example of a drug that's involved in clotting and reducing blood clots called Plavix. And some populations from the Pacific Islands, right? Many populations from the Pacific Islands, most people can't make use of that medication, right? So, if you prescribe them that medication, then they're not going to have a good response, right? And so you have to know that. But, that's one where again, you've got a good understanding of the genetics, you've got a good understanding of the trait. It becomes a question of getting people, it's like their blood type, right? Blood type, tends to be pretty universal. ABO blood group, every group has people with ABO blood group, Rh positive, Rh negative, but you still have to know those blood groups in order to do a transfusion, right? And other blood groups, right? So we're very interested in those questions. How do we get to what we would call the Galatea Bio Passport, right? What is the minimum set of things that you want everyone to carry? Kind of like old school, right? Going back to Venezuela. My dad used to have a little thing around his neck, the chain that had his blood group and
the medical alert bracelets. Yeah.
Like a medical alert bracelet.
Yeah, you got me thinking there, like on the, again, coming back to your median analogy. So for example, my earbuds that I wear, so I know that I'm, sensitive to noise induced hear loss, right? So I have a setting, I have an upper setting on my device, and I meet with my hearing doctor regularly, and there are things that I can do to control my environment nutritionally, my general health, et cetera. But it is actually pretty cool. Like what you're saying, your Galatea type passport, it's like you'd upload this DNA information and there'd be like all these equalizers around, well, the different drugs that you may or may not have to take, the different interventions that, and you can't even, you can't even ascribe it to, race or origin, because we're just this, like we said, this cosmopolitan genome, like there's all this mixing actually. And I did have, so I'm going to finish this with a question that, well, maybe I'll jump back to that one later. I think let's talk through like that Galatea Passport idea? Like is how far are we away from something like that?
Yeah. Well, we're working on aspects of it. Right? So the first thing we have in the passport is your ancestry. Right. So let's get to a really good ancestry painting and ancestry annotation that's based on whole genome sequence data. Right. So we can now go through and essentially color the chromosomes that come from your mom and from your dad based on whether they have a recent European, African, South Asian, Middle Eastern, etc. origin. And even within those, look more Spanish, Italian. Within Africa, they look like West Africa, South Africa, North Africa, etc. And then hang off of that, additional interpretation. So we've got a cardiometabolic panel that's coming. That has your type 1 diabetes, type 2 diabetes, your cholesterol risks, atrial fibrillation, strokes. And then we're working on cancer to deploy at the end of the year. And again, my view is the best genome is a whole genome. Right? We sequence the whole genome and then we sort of tranche it out, right? So cancer risk. If you have a BRCA1 or 2 pathogenic mutation, then we know you're in a high risk category. I don't really need to know what's in the rest of the genome, right? There's not a lot that I would say, oh, I'm going to attenuate it. No, that's what's going to drive decision making. But many people don't have a BRCA1 or 2 canonical breast cancer mutation. They might have one in one of the other 100+ genes that we know are involved in cancer. CHEK2, for example. Well, now you got to think, okay, maybe, right? Like maybe the rest of the genome attenuates me one way or another. Do I screen more regularly than not, et cetera? And we're doing this with cholesterol. So we have a very interesting project right now on people with very high bad cholesterol. And these samples are coming to us from all over, including from Mexico. And we're finding that these people who were sick enough with high cholesterol to be seen in the specialty clinics in Mexico, many of them have a known mutation in these canonical genes, but then the rest of the genome is also looking like it's loaded, right? Which kind of makes sense because it may not be, sufficient to have one of these mutations in a canonical gene in order to present with the phenotype. But if you present with a phenotype, then more often than not, then you may carry one of these mutations, right? So you had genopheno. You start with a phenotype and you're looking under the lamppost and you find the usual suspects, then great. Like we have guidelines and at least genomic medicine is beginning to build an edifice of care that we can begin to tranche people into. If you don't fit into what we already know, is it because you come from an underrepresented ethnic group? And so we need to do more to annotate and understand whether there's missing information, right? Kind of dark matter there that isn't represented. Or is it that it's the rest of the genome that is interacting in the way we as quantitative geneticists have always stipulated? Oh, there are no genes for, rather the whole genome is interacting with the rest of the genome and producing phenotype. And so we're getting to slice all those different layers of interpretation, right? So it's a signal to noise problem, so let's look first under the cleanest signal. If you can get the signal there, then you can follow and eventually, rising tide lifts all ships, right? So if we have a very diverse database and we've got lots of partners and we can accelerate learning and turn the crank on getting this faster, better, cheaper for everybody, right? So if I think about the technology that delivers, our ability to be on a podcast wirelessly and streaming free to the whole world, right? Like it's because of massive advances in lots of different allied parts of technology, semiconductors, uh,
Mm-hmm.
signal processing, transatlantic wiring,
Mm-hmm.
SpaceX,
Mm-hmm.
whatever, right? So you've really created a whole ecosystem where the marginal cost of this podcast is several cents to get out to people, and so you can give it away for free, right? So at what
Mm-hmm.
point can we give away for free genomic
Mm-hmm.
interpretation, genomic sequencing, and really put it in the hands of the masses? Well, at the time when you're entering almost what would be called the negative dollar genome, right? It's about eyeballs, and it's about getting people to help you annotate this information.
Mm. So you mentioned previously in the conversation, well, we've been talking around the direct to consumer genomics industry. And one of the things that you mentioned is, well, docs aren't educated on how to do the genomic, barely educated on how to do the genetic interpretation. We know we are under
Through no
resourced in
fault of
terms of,
their own. I don't want
no,
to
no, no, no.
blame the
Yeah, no. I mean,
doctor, right? I mean,
you can't, you're a
they're
son
also
of a
not
doctor.
trained to
Yeah.
change. They're also not trained to change motors or to
Yeah.
change the light bulbs in the
Yeah.
car.
Right. And so that wasn't an assault on docs. That was really more stating the problem. Like, even we know we're underrepresented in terms of genetic counselors. There aren't enough.
Oh, huge.
Yeah. Yeah. And so then, but all this information is coming, right?
Right.
And so
But
how
and the
how are we
operative
going
word,
to?
I'm a huge fan.
Yeah.
I'm a huge fan
Yeah.
of genetic counselors. I say it's a high
Yeah.
IQ, high EQ business.
Yeah. Yeah.
And
Big time.
the operative word in genetic counselor is counseling,
Mm.
right? It's counseling. It's the guide
Mm-hmm.
that helps you navigate, so it is somebody to sit there and help you understand your options, if there are options, because sometimes there's not options, right? There's maybe nothing
Right.
you can do, right? It's information you have to process. But if
Right.
there are options, what are they, and what's the evidence behind taking decision A versus decision B, which is often nerve wracking, right? Like, it's like
Yeah.
when you go to the doctor and the doctor says, well, you could take this medicine or that medicine, and which would you like to do? You say, well, you're the doctor. Tell me what to do, right?
Yeah.
But
Yeah.
that's not the way. At the end of the day, we have
Yeah.
to be stewards of our own health. We are tasked with, the doctor's there as a guide. Honestly,
Mm-hmm.
they're there
Mm-hmm.
to
Mm-hmm.
help us maintain health, and if we're sick, return to health, and if we're very sick,
Mm-hmm.
to put us in the hands of a doctor who may be a specialist, who may be able to help us in the sickest of times, right?
Right.
So
Right. Right.
um, they have a duty and a mission, almost a vocation, as it were, to uh, pastor to us in the shepherding way and shepherd us along. But ultimately, we all have to take responsibility here and take charge of how we make use of this information. The same way we take charge of the diet and exercise.
Right. Right.
You
So
know,
maybe we need to be introducing genetics, and I know we are. I think it's like in at least in high school now, if not elementary school. I mean, maybe that's when it's. I mean, computer coding has started, earlier. So maybe that's it. Is genomics is part of the conversation earlier rather than later. Here's a general question for you: 23andMe, did that help or hurt the industry?
oh, I think the world of 23andMe. Here
Mm-hmm.
is a woman who could do anything with her time and resources. Anything,
Mm-hmm. Mm-hmm.
right? She is blessed with a bountiful talent, bountiful resources, and bountiful connections,
Mm-hmm. Mm-hmm.
right? And she has chosen for the past nearly 20 years
Mm-hmm.
to put her money, her reputation, her time to get this in the hands of as many people as possible,
Mm-hmm.
right? It might have been super early when
Mm-hmm.
she launched in 2006, 2007. It might have been that, she, in the Silicon Valley enthusiasm, putting technology in the hands of people perhaps alienated the medical class, and, lots of people have done that in the past too, right? And you could falter for that, or you could say, well, what about the Apple Watch, right? Nobody asked their doctors about the permission to use the Apple Watch. And for a long time, there were Fitbits and other things, and some weren't as good, and blah, 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 right? But, I think overall, it has been, a massive nonprofit in that it has not made a profit and has greatly subsidized the learning of genetics by the general population. It's been sustained by the iron will of a very, very determined entrepreneur. So, I admire Anne and what she's done and what she continues to do. I do think it is outside the price point of many people. Many people don't have hundreds of dollars to spend on genetics. And so we have to figure out a way to get genetics. How do we give it away, right? How do we give it away in order to have it and to keep it, right? Because I think that's where we ultimately end up, And, I'll contrast it with Ancestry.com. I helped Ancestry DNA start their DNA business. And Ancestry, very well thought out business model because they're in an old business. Ancestry DNA is not in a new business. It's in the family Bible business, right? In the front of the family Bible was your genealogy. And so, since Genesis, we have the begats and who begat whom and who's the son of whom and who's the, like the genealogy of the Christ that starts two of the four gospels, right? They start with the genealogy of Jesus.
Right. That's right.
Yeah. And so we've cared a lot about genealogy. It's been an important part of human history. And Ancestry DNA began a next phase of how do we get people to understand who their ancestors were. That's important in many faiths, including the Mormon faith that is tied and has been tied to that business for a long time, through not revealing anything that people don't already know. So people have been subscribing to genealogy services time immemorial, right? And it used to be you would get, magazines in the mail, kind of like catalogs, and then it's AOL online type of business, and then they added this as the next phase of it, but it was a different track. It didn't. Recently they've re-added traits. It's been hard for them to do traits because oftentimes, genealogy and ancestry are a different hobby, as it were, than the trade and health part of the business.
Well, it's interesting. I didn't realize that you had spent time advising Ancestry DNA. So that you must have been back there in the days of like Ken Shaheen and John Pereira. Is that right?
So Ken Shaheen came to my office at Stanford and said, I've got this crazy idea. We're trying to get Ancestry.com to offer a DNA test. What do you think? I'm like, yeah, bro, that's a good idea. You should do that. How can I play with you guys? So, yeah. No, I'm a huge fan of it. Look, I did the first dog DNA test. So I did Wisdom. Back when I was at Cornell. So we did the Wisdom panel. And then, and then my postdoc, Adam Boyko, who overlapped with Sean Miles, and his brother, Ryan Boyko, Boyko brothers, launched Embark. And I've been a scientific advisor and board observer on the Embark team. So I am a full-throated, fully paid up member of the direct to consumer class, man. I really think this is knowledge is power, and people should have the opportunity to get access to DNA sequencing come hell or high water, right? Like, I think it is really important. And now, with great power comes great responsibility. You have to educate people. You got to put information out there that's accurate. Uh, there's lots of folks that overhype what their DNA test can do and can tell you, oh, here's, I can tell the future. Right? Well, predicting the future is a lot harder than predicting the past, right? So,
Well,
what degree can?
yeah. So what I was thinking, just as you said that, I was like, let's finish up here with the beginning. And the beginning was, in a way, a misstep or a misthought on the future. So we talked about Gould and Lewontin, and their, his, I would say, underestimate of, statistical genetics or population genetics. What are we missing here today? And who will stand on our shoulders, on your shoulders, in terms of what is going on with, Galatea and the 10 million.
Yeah.
Yeah.
Well, I think, as I like to say, the methylation risk score is the next stage after polygenic risk scores, right? Because you're getting closer to the phenotype. So really understanding your methylation pattern and methylation more broadly other omics is the right direction to travel, right? So
when you say methylation, you mean epigenetics.
Epigenetics. Yeah, epigenetics.
Yeah. Okay.
And, and I know you're a fan of the aging space and whether you can tell biological clocks and so on. I think it's a fascinating question. I do. But more broadly, I think, we will get to liquid biopsy and liquid biopsy for lots of things beyond cancer, right? So I would like to see a future where you can get routine sequencing or monitoring. I mean, you can call it sequence. The fact that it's sequencing is in some sense irrelevant. Sequencing is just, light microscopy, right? It's microscopy for PCR products of the genome. So if you can get there and monitor health using blood and other tissue samples, I think that's going to be unbelievably powerful and useful. Right? Like think about what we just lived through with the pandemic and how our lives were upended by whether or not you could use qPCR to detect viral particles, right? Like society hinged on the results of qPCR tests. I don't know if we're going to go back to a time in our lifetime like that. I mean, it's a little bit like the solar eclipse, right? Like the sun was darkened out and the land stood still as, but ultimately science explained it. And the earthquake. Yeah, exactly.
The earthquake and the curtain broke and now everybody can see into the temple. Yeah, right. Yeah, that's
there
right.
You go. There you go. There you go.
That's
at 3:00.
It's right. That's right. That's interesting. So I think there's a liquid biopsy, and then of course it's not just going to be one. It's going to be at multiple times for multiple reasons in the 100%. Context of
100%.
So now, so it's not only here's the community of all the DNA in the context of the whole world, but then there's your isolated community of cells in the context of other closely related community of cells. And so what community do you want to move yourself to? And so I think what you're saying is maybe there are the number of discrete steps to where you want to be are more only because they're more obvious. I'm thinking about a much more.
Yeah, I mean, look, if I want to monitor chronic kidney disease, I may be able to get to a blood test that includes DNA, it includes other biomarkers, right?
Right.
So,
right.
We have all, and here's what's interesting, right? Medicine has relied on pretty much the same set of samples for a couple hundred years.
Right.
It's urine, it's blood, it's feces, it's sputum, right? we haven't, I mean, lumbar puncture, right? But we didn't invent new tissues, right? We've just gotten better at imaging and reading entrails, uh, where we've been able to get to signal over noise that can manage treatment decisions. And look, whether or not you make the right treatment decision based on a reproducible test or not, that's like beyond the test maker's realm, right? Our job as testers is to get you a reliable, reproducible test, right? I can reliably sequence the genome. I can reliably find mutations. What you go do with that forward, that's an interpretation issue, and in some sense a little bit outside the hands of the diagnostician, right? But I do think we're going to narrow paths to successful treatments absolutely based on biomolecules, right? And then those biomolecules, you'll also be able to dial up or down using supplements, medications, behaviors, right? And ultimately extend not only lifespan, but health span, right? It isn't about just keeping people alive. It's about having really good quality and quality of life to people, right? We're going to get to cures. We're going to get to cures first for serious genetic disorders, and then for other conditions that have a genetic entry point to understand the biology. I mean, that's the logic of it. It's not just genetics for genetics' sake, but rather I can use the genetics to understand the biology and mechanism, and then drive treatment based on mechanism, because it should be rational, right? It shouldn't be chicken entrails and divination bones, right? And then ultimately about universality of practice based on protocolized treatments, right? Because it doesn't help if you get to the world's best treatments, but they're not in the hands of physicians and they don't understand it, and then it just dies on the vine, right? You don't want that.
So you've been going 3 years. You're already close to 1 million samples. You're probably going to be at 10 within, the next 3 years. I would
Something
guess
like that.
Yeah. How do people sign up to become, a part of your, it's not just a research cohort. I mean, a better way to say it is, how do people sign up to understand their biology? Where do they find you?
Awesome. There's kind of three ways. The first is through our protocol, Biobank of the Americas. This is free to participate. bbofa.org. You can sign up. We'll send you a kit, and we'll get you sequenced. And that's kind of the equivalent of the over area intent, right? We'll give you that as part of contributing to science, sort of the citizen science. Right? Then, secondly, we have some premium tools that are now we hope competitively priced, and so we're working with partners like you guys and sequencing.com. So if you'd like to start with your ancestry or your cardiometabolic panel and so on, if you have data, bring your data. We'll give you an interpretation. If you don't have data, we'll collect data for you. And so that's sort of a second. And then third, if you happen to be part of a community with a necessity for genetic testing, let's put it that way. If you meet criteria for genetic testing, then you can come to Galatea Bio. We have a clinical lab. We can get you sequenced and your genome interpreted in light of conditions that you may be presenting with. And we're very interested in Mendelian conditions and complex disease and autism and the whole spectrum. And so you can reach out to us at info@galatea.bio, and then we can work with you and your physician to get you worked up on a clinical basis. So there's the freemium, There's the direct to consumer with partners like you guys, and then there's the more clinical path forward. And in some cases, insurance could cover it. In other cases, we'll work with you and your doctor to get you sequenced if it merits criteria, if you meet criteria for sequencing.
Amazing. And what about you? What about Dr. Carlos Bustamante? Where do people find you? Are you like LinkedIn? Are you Twitter or X? Where do they find you?
I'm on LinkedIn. I'm on Twitter. We'll put those in the news feed here.
Absolutely.
Bustamante is my my Twitter handle. You can also find me on LinkedIn. And again, if you drop us an email, info@galatea.bio. I monitor that. We have a whole team that monitors that, and we'll try to get to you. One of the things I always tried to do when I was running the Stanford lab, we got lots of letters from people who wanted certain kinds of genetic testing, ancestry testing. For example, that's why we spun out this ancestry business, spun it up so that we can give you the ancestry test that we promised. And we're always interested in working with people, with academic partners, with rare disease groups, and so Galatea Bio is here to serve. So thank you for the opportunity to connect, Jeremy. It's always a pleasure to talk to you, and love the pod and what you're doing. So congratulations on over a dozen episodes in. I hope this is like Lex Friedman, right? You can have interview 2, at some point in the future.
I think so. I think so. I've got like 1,000 more questions I want to ask you. I got to say, Dr. Carlos Bustamante, this distinguished American. What's that?
I said we could do the next one on dog genetics and horse genetics and plant
game
genetics,
on.
whatever you want.
Oh yeah, game on. Well, a distinguished American population, I should say Venezuelan American population geneticist as yourself, academic entrepreneur, currently CEO of Galatea Bio. Thank you so much for joining, and I'm looking forward to that next conversation.
My pleasure, brother.
Thank you for tuning in to the DNA of Things. Today's conversation with Dr. Carlos Bustamante offered an enlightening look at the intersection of genetics, evolution, and data science. You probably sensed his passion for population genetics and his leadership at Galatea and how they are paving the way for a future where medicine is not just personalized, but it's inclusive. We delved into the challenges of creating diverse genetic databases, the development of the Galatea Bio Passport, and the critical role that genetic counselors play in navigating this new terrain. Dr. Bustamante's work reminds us that in the tapestry of human genetics, every thread counts, highlighting the importance of representing all ethnic groups in genomic studies. The journey towards a more precise and accessible genomic medicine is long, but the pioneers that we have working for us and towards this direction, like Dr. Bustamante, makes this future look promising. So stay curious, folks, stay informed, and join us next time as we continue to explore the DNA of everything around us. Remember, understanding our genetic makeup is the key to unlocking a healthier future for all.